Hello, my name is Taylor Thompson, and it is July 7th, 2024, and I am doing a presentation on anatomy and physiology of the retina. It is for the University of New England online, Physiology 1010-06E. I'm sorry, in the background you will hear my dog chewing on a bone. This is to placate her while I'm doing this because she wants to play very badly right now. So with that, we will start with steps of the vision pathway. So the vision pathway can be summed up in 3 steps, more or less. First, it's light from the environment enters the cornea, and so you can see that in our first image here. Next, phototransduction occurs at the retina. That would be our second image here. And third, processing of light signals by the brain, and that would be our final image down here. Next, we're going to start with our fundamentals, which is always the fun stuff, definitions. I will be jumping between this slide and the previous one, just to demonstrate where they are in relation to the images to better help visualize this stuff. So our retina, that would be the layer that contains the photoreceptors. So jumping back, that would be this whole encompassed picture, but looking at this picture, it would be that yellow thin line going about 2/3 away around the back of the eye. Next, we have our photoreceptor cells, and these are our neurons that convert light energy into electrical signals, also known as our action potentials. There are 5 types of neurons. We have our rods and cones, which make up 2 of our main types of photoreceptors. Our rods, they function well in low light. They're specifically for night vision, and our cones are responsible for vision and color, or high activity vision and color vision during the daytime. So in this second image, you can see along here we have our rods and cones. Cones are depicted as purple and yellow are our rods. We have our ganglion cells, and these are neurons on the eye whose axons form the optic nerve. So our ganglion cells are these blue cells, and they come together and form this thick yellow part known as our optic nerve. Next, we have our horizontal cells. These adapt to both bright light and dim light conditions, and they're providing feedback to our rods and cones. They're not depicted on this image, but they will be in images to come. Next, we have our bipolar cells. They provide the main pathway for our photoreceptors to our ganglion cells. So in this image, they would be these red cells, and you can see they're connecting our rods and cones to our blue cells, which are the ganglion cells. Finally, we have our amacrine cells. They receive synaptic input from our bipolar cells and other amacrine cells, and they provide input to other amacrine cells and our ganglion cells and feedback to the bipolar cells. They're not depicted on here, but they will be in future images. Next, we have phototransduction. So that encompasses this entire process of light hitting the retina, and then creating an action potential. Our optic nerve, so that's what's transmitting our electrical signals or our action potentials to the brain from the retina. So this action potential is created, goes along here, along our ganglion cells to our optic nerve. And so our optic nerve is comprised of all of these ganglion cells. Next is rhodopsin, and that is the visual pigment of our rods, which we will go into further later on. Next, so this is the first step in the vision pathway. So light enters through the cornea. So that would be this layer, white layer right here. And it is modified in 2 ways. First, the pupil contracts or dilates to control the amount of light that's entering and reaches the photoreceptors. And the second, the light is focused by changes in the shape of the lens. So what this is meaning is that it refracts. Next, the light strikes the lens, which would be this big white thing here. And it has two convex surfaces to focus light on the retina. The light is actually refracted twice, first through the cornea and then again through the lens to focus it on the photoreceptors. Next, once light has entered the eye, this is where our phototransduction occurs at the retina. Now, this image contains a lot of verbiage, and this is just to summarize the process if you are the type of person who needs to read this to understand. I'm going to go through it here through these images. So first, big picture, this is, as earlier shown, our optic nerve right here. So our light would be entering, and this image shows light entering, hitting our photoreceptors right here, so our rods and cones. And then from there, this next image nicely shows our rod, in this case, our bipolar cell, our ganglion cell, our pigment epithelium. And finally, this picture finally shows our horizontal cell and our amacrine cells as well. So you can see our horizontal cells that provide feedback between the rods and cones are these little light blue ones, and then our little round ones are our amacrine cells. So our light hits those cells. You would think, or at least when I first was learning this material, I thought it hit these blue cells, and then that our action potential somehow got created and went along the pigment epithelium. But it's actually the reverse. It hits the back area, and then we actually move in this direction from the rods, the bipolar cells, to the ganglion cell, and then to the optic nerve. And so when light hits, whether it's in light or darkness, that determines our action potential. So first we have our, I'm sorry, hold on one second, my dog is upset. So, sorry about that. So, in darkness, this image summarizes very well that we don't have light hitting our rhodopsin right here. And so we still have and our cGMP levels remain high. And so we have calcium, sodium with a high influx into the cell and potassium with a low efflux out of the cell. So this creates depolarization. And with that, this causes glutamate neurotransmitters to release, causing a feedback loop. So no action potential is created. But if we have light hitting our rhodopsin, it separates, our activated retinal separates from it and is taken up by the pigment epithelium. And this causes a cascade where we decrease our cGMP, which then actually closes our calcium and sodium channel. So we no longer have an influx of calcium or sodium, but we still have our potassium leaving the cell. So we have a hyperpolarization, which causes our neurotransmitters, glutamate, to no longer release, or if they do, it's in very small amounts. So then this creates our action potential, and our action potential is then followed down through the bipolar cell, the ganglion cell, into our optic nerve. Next, the processing of light from the brain. So we have our optic nerve, and that is comprised of a bunch of ganglion cells. We have millions of them in our eyes, each eye. And so these action potentials follow down the optic nerve, and then we come to our optic chiasm. The optic chiasm, some of the nerves decussate, and they actually go on for control of eye movement and coordination with the somatosensory and the auditory information for balance and movement. But most of them continue on our optic tract to our thalamus, and also known as our lateral geniculate body. And this is where the optic fibers synapse onto neurons, where they lead to the visual cortex and the occipital lobe. Finally, here are my references for the images. And then as my student worksheet, I kind of like to get into arts and crafts a little bit, and just to take my mind away from reading for a bit and do some more motor neuron work. I actually drew up a sun for hitting an eye, the light. And then I have my bipolar cells to then connect to my my ganglion cell to connect to my horizontal cells, and then to connect to my cone, that little cone here. And then I also have my pigment epithelium to connect, and then I also have my rods. And then additionally, I have labels to label everything. So this one says horizontal cell. We have our amacrine cell, bipolar cell, cone. And so I just wanted to be able to do this kind of like a labeling, but make it a little more arts and craftsy. Thank you so much, and have a good evening.